Genetic mutations. Mutations are changes that occur in genetic material DNA and RNA, but at the gene sequence level called mutations dots, as well as at the chromosomal level. Mutations at level chromosomal aberrations are usually called aberrations. Mutations in genes can on the emergence of new alleles and become the basis that leads to evolutionary advocates regarding the emergence of variations new to the species. The term mutations was first used by Hugo de Vries to suggest a phenotypic change on Oneathera lamarckiana flowers and is hereditary. It turns out the change occurred due to deviations from the chromosome. Seth Reich also reported mutations in Anton sheepbread, which is short-legged and hereditary. Scientific research on mutations was also carried out by Morgan using Drosophila melanogaster. Gain mutations are basically point mutations. In this mutation, there is a chemical change in on or multiple base pairs in a single can cause a change in individual characteristic without a change in quantity and the arrangement of the chromosome. Events that occur in mutations gains are changes in DNA sequence or more precisely a point mutations is a change in the N base and DNA or RNA chromosomal mutations also known as mutations large or gross mutation or chromosomal aberrations is a change the number of chromosome and the structure of genes in chromosome. Chromosomal mutation often occur by mistake meiosis and less in mitosis. Cause of mutation. Chemical mutagens example are colchicine and digitonin substance. Colchicine is a substance which can prevent the formation of spindle fibers in the anapase process and can inhibit cell division in anapase. The cause of mutations in the environment that are chemical in nature are called also chemical mutagens. Mutagen material P6 example of physical material mutagen are ultraviolet light like radioactivity and gamma rays. Biological material mutagen, viruses and bacteria are thought to cause mutations. The part of the virus that can cause mutation is his DNA. Kinds of mutations. Mutations type gene mutations. 1. Based on the incident. Spontaneous mutations and induced mutations. 2. Based on the type of mutate, mutation cell. Somatic mutation and germinal gametic mutations. 3. Based on change in the genetic code. Omissions mutations, silent mutations, substitution, nonsense mutation, and from shift mutations. Types of chromosomal mutations in gamete cell. 1. Autosomal mutation sex cell mutations that occur on autosomal chromosome. Mutation of the type produce dominant mutations and recessive mutation. 2. Sex linked mutation sex cell mutations that occur on the sex chromosomes in the form is of linking of several genes in the sex chromosome. Article about mutations. The mutation spectrum of the ATP7B gene in Vietnam's pediatric patients with Wilson disease is both diverse and distinct from of other Asian populations. This once again confirms that mutational hotspots in ATP7B Gain reports considerably vary by geographic regions. Based on these results, the study developed a streamlined procedure to quickly characterize mutations in the ATP7B gene in the Vietnam children with VD. This procedure plays a vital role in diagnosis, and it will make genetic analysis for clinically substate patients quicker in order to detect disease earlier and restrict complications of the WD disease. In addition, novel and pathogenic variants reported in this study will be significantly added to the archive of ATP7B gain bank. The common mutations and hotspot regions of ATP7B gain in Vietnam pediatric patients are also very helpful for screening asymptomatic sibling of VD patients. Thank you.